Good day, sir. I'm Pavitra and Aisha will be presenting about our bioinformatic project on the silico structural and functional annotation of a bacteria hypothetical protein from Acinetobacter baumannii. This study is driven by three key main objectives. First is to identify the structural characteristics of this protein by locating its structural domains, conserve motif, and candidate active sites using computational pipelines. Second, to infer its biological, functional, and possible metabolic pathway involvement via orthology mapping and structural homology modeling. Third, to evaluate its evolutionary conservation across bacterial species to establish sequence, structures, and function relationship. So for the introduction, bacterial genome sequencing continuously generates open reading frames faster than they can be experimentally characterized, leaving a vast share classified as hypothetical proteins. Computational pipelines allow us to infer biological function in silico before committing to coarse wet lab experiments. At a glance, our target protein is WP underscore 0000552501. It consists of 429 amino acids with a calculated molecular weight of 40.4 kDa. It currently has 0% created representation in Swiss prot and lacks confirmed functions. So for the tools and database used is we utilize a pipeline of 24 publicly available surf and database. These were categorized across sequence and evolutionary analysis and physiochemical characterization, subcellular location and topology, domain and fall prediction, 3D structure prediction, and model validation using tools like SAFES V6.1 and CASP. As for evolution, starting with sequence similarity, the BLAST search yield top hits with 99.77% to 100% identified. Crucially, all top hits belong exclusively to Acinetobacter baumannii, a multi-drug resistant non-social pathogen. The absence of significant hits in uniprot or Swiss prot confirm that this protein is truly experimental, uncharacterized. Multiple sequence alignment showed near total conservation across sequence, while the biological tree displayed a tight clade with very strong branch lengths, pointing to strong purifying selection and indicating a functional important proteins. So for the result, physiochemical properties using XPASI prot-pram, we establish its physical and chemical properties. The protein has an isoelectric point of 5.62, showing an acidic character and instability index of 55.51, classifying it as an unstable in vitro, and a negative gravity score of negative 0.372. Together, the acidic PL and negative gravity score predict a soluble hydrophilic cytoplasmic protein. And also for the location and topology, this cytoplasmic prediction was overwhelmingly confirmed across 4 independent subcellular location tools. Will be like Cello, Pisoptop, Sociogram, and PSL Pret. Furthermore, TMH, MM, and CCTOP topology models predicted zero transmembrane helices, confirming that WP 000552501 is indeed a soluble non-membrane protein. I will pass to Aisha.
So I'll be talking about the domains, motif, and fold. So for functional annotation, the NC by CD research ultimately came back empty. So no significant domain hits. So we looked at like other databases. PFAM gave us 2 weak matches, IP1 and also DUF1040. And but the E value was like 0.036 and 0.049. So are both like above the significant cutoff. So we are not leaning on those. So the strong result came from the superfamily, a thioredoxin-like superfamily match at residue 241 to 422. So with an e-value of 8.27, 10 times 10 to the minus 9. So extremely significant. And superfamily also offered a more specific DSBA like family label, but that's one e-value was like only 0.056. So we are like deliberately not claiming that narrow label, just like the border thioredoxin 4. And then psi-PRED predicts the protein is mostly like alpha helical, 56.7%, with the rest split between random coil and a small amount of beta strands. So for the 3D structure, 5.2 model is against a glutathione S transferase template at 99.6% confidence. But it only covered like 42% of the sequence, 182 out of the 429 residues. And when we validate that model, ERAT, verify 3D and also Ramachandran flow. So all three scores came in below the usual benchmark for a high quality structure. So our honest takeaway is that this is like a useful, moderate confidence partial model for building a hypothesis and not like a finished structure. And then Strength predicted two functional partners for our proteins with confidence scores of 0.693 and also 0.573. So what interesting is that our proteins on locus tag A1S_3454 sits numerically right between them. So which is like a bacterial genomes often hints at a shared gene neighborhood. So the overall network wasn't statistically significant on its own, like p-value 0.527, but a more a specific enrichment did come back significant. So an antioxidant related cluster involving cytochrome B561 and catalases, so which actually fits nicely with the thioredoxin fold idea from the last slide. And last, for the active site, CASP was down the entire time we tried. So we don't have an area or like a volume data. So we're reporting that honestly as a gap rather than like a guessing. And then here's how we connected everything together. So two completely independent methods point to the same region of the protein: superfamily sequence-based classification and the 5-2 structure-based modeling. So both land on the thyrodoxin relative fold around residue 241 to 422. So our working hypothesis is that this C-terminal region has a thyrodoxin-like fold, possibly involved in oxidoreductase or disulfide related activity. And we're intentionally not naming a more specific family since the supporting evidence for that, the DSBA call, the string enrichment is suggested and not conclusive on its own. A few ethical points worth raising is that firstly, everything here comes from public databases and third-party tools, none of it's our own. So proper citation matters. And secondly, there are computation prediction, not experimental proof. So we try to always report since Acinetobacter baumannii is a real pathogen. This kind of annotation would support drug discovery. Thirdly, but the same technique do carry a general dual use consideration that researchers should be aware of. Okay, to sum up, WP_0000552550.1 is a highly conserved cytoplasmic protein in Acinetobacter baumannii, and our evidence point towards a thioredoxin 4, possibly oxidoreductase related domain, though that's still a computational hypothesis and not a confirmed functional. So for future work, we had recommend recombinant expression and structure verification by X-ray or cryo-EM, targeted enzyme assays, gene knockouts, or CRISPR testing for essentiality, and closer to home rerunning CASP to finally get the active site data we are missing. So that's all for our full analysis. Thank you.